This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Introduction. Welcome, science enthusiasts and curious minds. Today, we're diving into the fascinating world of turtle evolution. Turtles have captured our imagination for centuries with their unique body plan and remarkable adaptations. In this video, we'll explore recent scientific research that sheds light on the evolutionary journey of these incredible creatures. From their mysterious origins to their diverse modern forms, turtles have undergone an extraordinary transformation over millions of years. We'll uncover the secrets hidden in fossils, examine the development of their iconic shells, and discover how these resilient animals have adapted to life on land, in freshwater, and even in the vast oceans. Keeping up with scientific literature is crucial for both professionals and enthusiasts alike, as it allows us to deepen our understanding of the natural world and appreciate the complexity of life on Earth. By the end of this video, you'll have gained insights into the evolutionary processes that shaped turtles, the evidence supporting current theories, and the broader implications of this research for our understanding of evolution as a whole. So, settle in and prepare to be amazed by the evolutionary tale of turtles, a story that spans over 220 million years and continues to unfold before our eyes. Let's begin our journey through time and discover how these remarkable creatures came to be. 1. Origin and early evolution of the turtle body plan. The origin of turtles has long been a subject of fascination and debate among scientists. Recent research has provided exciting new insights into the early evolution of the turtle body plan, pushing back the timeline of their emergence and revealing surprising details about their ancestral forms. The turtle lineage is now believed to have diverged from other reptiles during the Permian period, approximately 260 million years ago. This places the origin of turtles even before the rise of the dinosaurs, making them one of the oldest surviving groups of reptiles on Earth. One of the key discoveries in recent years has been the identification of several early turtle-like reptiles that help bridge the gap between more conventional reptilian forms and the unique body plan of modern turtles. These transitional fossils have allowed researchers to piece together the gradual evolution of turtle characteristics over millions of years. One such transitional form is a species called Eunotosaurus africanus, which lived around 260 million years ago in what is now South Africa. This ancient reptile had several features that hint at its role in turtle evolution, including broadened ribs and a boxy body shape. While it lacked a fully formed shell, the expanded ribs of Eunotosaurus suggest the beginnings of the turtle's characteristic carapace. Another crucial transitional form is Popochelys rozanae, nicknamed the grandfather turtle. This 20-centimeter-long reptile lived about 240 million years ago in what is now Germany. Popochelys had significantly enlarged and flattened ribs, as well as other skeletal features that place it firmly on the path to becoming a turtle. Interestingly, both Eunotosaurus and Popochelys were terrestrial animals capable of digging, suggesting that the early stages of turtle evolution may have been closely tied to a burrowing lifestyle. The discovery of these transitional forms has helped resolve a long-standing debate about the evolutionary relationships of turtles to other reptiles. The skull structure of Popochelys, in particular, shows characteristics typical of diapsid reptiles, indicating that turtles are more closely related to modern lizards and snakes than to other extinct groups of reptiles. One of the most intriguing aspects of early turtle evolution is the development of their unique breathing mechanism. Unlike most reptiles, which use rib movements to breathe, turtles have a specialized muscular system that allows them to breathe despite having rigid rib cages. Research suggests that this novel breathing mechanism may have evolved in response to the gradual stiffening of the rib cage in early turtle ancestors. The evolution of the turtle body plan also involved significant changes to the shoulder girdle. In most reptiles, the shoulder blades are located outside the rib cage. However, in turtles, the shoulder blades are found inside the ribs, a unique arrangement that required a major reorganization of the skeletal structure during evolution. These early stages of turtle evolution demonstrate the power of incremental change over long periods. What began as slight modifications to the ribs and body shape in animals like Unodontosaurus eventually led to the highly specialized and instantly recognizable form of modern turtles. This evolutionary journey highlights the remarkable adaptability of life and the diverse paths that evolution can take in response to environmental pressures and opportunities. Understanding the origin and early evolution of the turtle body plan not only satisfies our curiosity about these beloved creatures but also provides valuable insights into broader evolutionary processes. It demonstrates how major anatomical innovations can arise through a series of smaller changes over time, and how the demands of different lifestyles can shape the course of evolution. As we continue to unearth new fossils and apply advanced analytical techniques, our understanding of turtle origins will undoubtedly become even clearer. Each discovery brings us closer to fully appreciating the remarkable evolutionary journey that led to the turtles we know and love today. 2. Fossil evidence and transitional forms in turtle evolution. The fossil record plays a crucial role in our understanding of turtle evolution, providing tangible evidence of the changes that have occurred over millions of years. Recent discoveries have dramatically expanded our knowledge of turtle ancestry and the transitional forms that bridge the gap between early reptiles and modern turtles. One of the most significant fossil finds in recent years is Odontochelys semitstatia, a 220-million-year-old species discovered in China. This remarkable fossil, whose name means toothed turtle with a half shell, represents a crucial transitional form in turtle evolution. Odontochelys had a fully formed plastron, the bottom part of the shell, but only a partial carapace, the top part, consisting of widened ribs without the bony plates seen in later turtles. This finding suggests that the bottom shell evolved before the top shell, possibly as a defense against predators attacking from below in an aquatic environment. The discovery of Odontochelys pushed back the known origin of turtles by about 10 million years and provided valuable insights into the step-by-step evolution of the turtle shell. It also sparked debate about whether early turtles were primarily aquatic or terrestrial, as Odontochelys was found in marine deposits. Another important transitional form is Progonochelys quenstedii, a turtle that lived about 210 million years ago. Progonochelys had a fully formed shell, both top and bottom, but retained several primitive features not seen in modern turtles. These included a long tail, a skull with small openings behind the eyes, and teeth on the roof of its mouth. The presence of these features in Progonochelys helps illustrate the gradual nature of turtle evolution, showing how characteristics were acquired or lost over time. Fossil discoveries have also shed light on the diversity of ancient turtles and their adaptations to different environments. For example, the remains of giant sea turtles like Archelon, which lived during the Cretaceous period, demonstrate that marine turtles evolved to enormous sizes in prehistoric seas. Archelon could reach lengths of up to 4.6 meters, dwarfing even the largest modern sea turtles. In recent years, paleontologists have also uncovered fossils that provide insights into the evolution of specific turtle groups. For instance, the discovery of ancient side-necked turtles in North Africa has helped clarify the origins and dispersal patterns of this group, which is now found primarily in the southern hemisphere. The fossil record has been particularly valuable in understanding the evolution of turtle shells. By examining fossils from different time periods, researchers have been able to trace the gradual transformation of ribs into the complex, interlocking structure of the modern turtle shell. This evolutionary sequence begins with the broadened ribs seen in early forms like Unodontosaurus, progresses through the half-shell stage exemplified by Odontochelys, and culminates in the fully enclosed shells of Progonochelys and its descendants. One of the challenges in studying turtle evolution through fossils is the relative rarity of well-preserved specimens. Turtles' aquatic habits and the delicate nature of their shells can make fossilization difficult. However, exceptional fossil deposits, such as those found in ancient lake beds or marine environments with rapid sedimentation, have yielded beautifully preserved turtle fossils that provide a wealth of anatomical details. Advanced imaging techniques have revolutionized the study of turtle fossils in recent years. CT scanning, for example, allows researchers to examine the internal structures of fossilized turtles without damaging the specimens. This has led to new insights into the evolution of turtle skulls, limbs, and other anatomical features that are not always visible from the outside. The fossil record of turtles also provides valuable information about past climates and environments. The presence of turtle fossils in a particular location can indicate warm, aquatic habitats in the distant past, even in areas that are now arid or cold. This makes turtle fossils important tools for paleoclimatologists studying long-term climate change. As new fossil discoveries continue to be made, our understanding of turtle evolution becomes increasingly refined. Each new find has the potential to fill gaps in our knowledge, challenge existing theories, or provide unexpected insights into the evolutionary history of these fascinating creatures. The study of turtle fossils not only illuminates the specific evolutionary path of this group but also contributes to our broader understanding of how life on Earth has changed and adapted over hundreds of millions of years. 3. Development and evolution of the turtle shell. The turtle shell is arguably one of the most distinctive features in the animal kingdom, and its evolution represents a remarkable example of how major anatomical innovations can arise. Recent research combining fossil evidence, comparative anatomy, and developmental biology has provided fascinating insights into how this unique structure evolved and continues to develop in modern turtles. The turtle shell consists of two main parts: the dorsal carapace, top shell, and the ventral plastron, bottom shell. What makes the turtle shell so unique is that it incorporates parts of the skeleton that in other animals remain separate, including the ribs, vertebrae, and shoulder girdle. This integration of external armor with internal skeletal elements is unparalleled among vertebrates and has long puzzled evolutionary biologists. Developmental studies of modern turtle embryos have been crucial in understanding how the shell forms during an individual turtle's growth. As a turtle embryo develops, its ribs begin to grow outward and broaden, eventually fusing with bony plates in the skin called osteoderms. This process creates the carapace, while the plastron develops from the clavicles, interclavicle, and abdominal ribs. The entire process is controlled by a complex interplay of genes and signaling molecules. One of the key genetic players in shell development is a gene called WNT5A. This gene is expressed along the edges of the developing carapace and helps to direct the growth of the shell. Other important genes include those in the FGF, fibroblast growth factor, family, which are involved in the broadening of the ribs and the formation of the bony plates. The evolutionary history of the shell, as revealed by the fossil record, shows that this complex structure developed gradually over millions of years. The broadened ribs seen in early turtle ancestors like Eunotosaurus represent the first step in this process. Over time, these ribs became even wider and began to fuse with bony plates in the skin, eventually forming a complete carapace. Interestingly, the plastron seems to have evolved before the carapace was fully formed. This is evidenced by fossils like Odontocheles, which had a complete plastron but only a partial carapace. This suggests that the evolutionary pressures driving the development of the shell may have initially come from below, possibly as a defense against predators in aquatic environments. The evolution of the turtle shell required not only changes to the skeleton but also significant modifications to the muscular system. In most vertebrates, the muscles that control breathing are attached to the ribs. However, with the ribs incorporated into the shell, turtles had to evolve a new way to breathe. This led to the development of a unique muscular sling that allows turtles to move air in and out of their lungs despite their rigid shells. Another fascinating aspect of shell evolution is the diversity of forms it has taken in different turtle lineages. Some turtles, like softshell turtles, have reduced the bony elements of their shells and developed a more leathery covering. Others, like sea turtles, have streamlined their shells for more efficient swimming. Box turtles have evolved hinges in their plastrons that allow them to completely close their shells for protection. Recent research has also shed light on the evolutionary plasticity of the turtle shell. Studies of fossil and living turtles have shown that the number and arrangement of bones in the shell can vary between species and even between individuals of the same species. This variability suggests that the turtle shell, despite its apparent rigidity, is actually a highly adaptable structure that can evolve in response to different environmental pressures. The development and evolution of the turtle shell also has implications for our understanding of evolutionary developmental biology more broadly. It provides a striking example of how existing structures, in this case, ribs and vertebrae, can be co-opted for new functions through relatively small changes in developmental pathways. Understanding shell development in turtles may also have practical applications. For example, insights gained from studying how turtles form their protective armor could potentially be applied in the development of new protective materials or in regenerative medicine. As research continues, we are likely to gain even more insights into the complex processes that led to the evolution of the turtle shell. This ongoing work not only enhances our appreciation for these remarkable creatures but also contributes to our broader understanding of evolutionary processes and the incredible diversity of life on Earth. 4. Adaptations to different habitats: terrestrial, freshwater, marine. Turtles have successfully colonized a wide range of habitats, from arid deserts to tropical rainforests, and from freshwater ponds to the open ocean. This remarkable ecological diversity is the result of millions of years of evolution, during which turtles have developed an array of adaptations to suit their various environments. Recent research has provided fascinating insights into how turtles have evolved to thrive in terrestrial, freshwater, and marine habitats. Terrestrial turtles, including tortoises, have evolved a number of adaptations for life on land. One of the most obvious is their high domed shells, which provide protection against predators and help prevent overturning. The legs of terrestrial turtles are typically sturdy and elephant-like, well-suited for supporting their weight on land. Many terrestrial species have also developed specialized digestive systems to handle a diet high in plant material, which is often tougher and less nutritious than animal prey. In arid environments, terrestrial turtles have evolved remarkable adaptations for water conservation. Some species can store water in their bladders or in specialized sacs in their body, allowing them to survive long periods without access to fresh water. The skin of desert-dwelling turtles is often thicker and less permeable than that of their moisture-loving relatives, reducing water loss through evaporation. Freshwater turtles, on the other hand, have evolved features that allow them to excel in aquatic environments while still retaining the ability to move on land. Many freshwater species have flattened, streamlined shells that reduce drag while swimming. Their feet are often webbed, providing greater propulsion in water. Some freshwater turtles have developed the ability to absorb oxygen through their skin or the lining of their mouths, allowing them to stay underwater for extended periods. One fascinating adaptation seen in some freshwater turtles is the ability to survive in oxygen-poor environments. For example, some species can lower their metabolic rate dramatically when oxygen is scarce, entering a state of suspended animation until conditions improve. Others have evolved specialized structures in their cloacas that function like gills, extracting oxygen from the water and allowing the turtles to stay submerged for months at a time. Marine turtles represent another remarkable evolutionary trajectory. These turtles have become highly specialized for life in the open ocean, with paddle-like flippers instead of feet, streamlined shells, and the ability to excrete excess salt through special glands near their eyes. The shells of sea turtles are typically flatter and more hydrodynamic than those of their terrestrial or freshwater relatives, reducing drag as they swim through the water. One of the most impressive adaptations of sea turtles is their ability to undertake long-distance migrations. Some species travel thousands of kilometers between feeding and nesting grounds, navigating using a combination of magnetic sensing, visual cues, and possibly even olfactory signals. The evolution of these navigational abilities represents a remarkable example of how turtles have adapted to the challenges of their marine environment. Recent research has also revealed fascinating adaptations in the reproductive strategies of turtles from different habitats. For example, many terrestrial and freshwater turtles have evolved temperature-dependent sex determination, where the temperature of the nest during incubation determines the sex of the hatchlings. This system allows for flexibility in sex ratios depending on environmental conditions. Marine turtles, on the other hand, often produce very large clutches of eggs to compensate for the high mortality rates of hatchlings in the ocean environment. The transition between these different habitats has occurred multiple times in turtle evolution, and recent studies have shed light on how these transitions may have occurred. For example, the ancestors of sea turtles are thought to have been freshwater turtles that gradually adapted to more saline environments, eventually becoming fully marine. This transition involved not only physical adaptations but also physiological changes to deal with the challenges of saltwater, such as the evolution of salt-excreting glands. Interestingly, some turtles have evolved the ability to move between different habitats. For instance, certain freshwater turtles can tolerate brackish water, allowing them to inhabit estuaries and coastal areas. This flexibility may provide insights into how ancient turtles made the transition from freshwater to marine environments. The study of turtle adaptations to different habitats has implications beyond just understanding turtle biology. It provides valuable insights into how animals can evolve to cope with diverse environmental challenges. This research can inform conservation efforts, helping us understand how turtles might respond to habitat changes caused by climate change or human activities. Furthermore, the adaptations of turtles to different habitats have inspired various fields of research and technology. For example, the hydrodynamic properties of sea turtle shells have been studied to improve the design of underwater vehicles. The water conservation strategies of desert tortoises have provided insights for developing more efficient water storage systems in arid regions. As we continue to study the diverse adaptations of turtles to their various habitats, we gain a deeper appreciation for the incredible plasticity of life and the power of evolutionary processes to shape organisms to fit their environments. Each turtle species tells a unique story of adaptation and survival, contributing to our understanding of evolution as a whole. 5. Phylogenetic relationships and classification of turtles. The study of turtle phylogeny, the evolutionary relationships between different turtle groups, has undergone significant advances in recent years, thanks to new fossil discoveries, molecular genetic techniques, and sophisticated analytical methods. This research has not only clarified the relationships between different turtle lineages but has also provided insights into the timing and patterns of turtle diversification. Traditionally, turtles were divided into two major groups based on how they retract their necks: Cryptodira, which retract their necks straight back into their shells, and Ploridira, which fold their necks sideways. While this basic division is still recognized, our understanding of the relationships within these groups and the position of turtles relative to other reptiles has become much more nuanced. One of the most significant developments in turtle phylogenetics has been the use of molecular data to complement morphological studies. DNA sequencing has allowed researchers to compare genetic similarities between different turtle species, providing a new line of evidence for evolutionary relationships. These molecular studies have largely confirmed the split between Cryptodira and Ploridira, suggesting that this division occurred very early in turtle evolution, possibly as far back as the Triassic period. Within the Cryptodira, molecular studies have helped to clarify the relationships between various families. For example, they have confirmed the close relationship between sea turtles, Cheloneoidea, and snapping turtles, Chelydridae, a connection that was not always clear from morphological data alone. These studies have also helped to resolve the positions of some enigmatic groups, such as the big-headed turtle, Platystron and Megacephalum, which is now generally considered to be closely related to the pond turtles, Emididae. Ploridira, while less diverse than Cryptodira, has also seen revisions in its classification based on molecular data. The three living families of side-necked turtles, Chelidae, Palomedusidae, and Podocnemididae, have been confirmed as distinct lineages, with Chelidae diverging first from the common ancestor of Ploridira. One of the most debated aspects of turtle phylogenetics has been the position of turtles within the larger reptile tree. For many years, turtles were thought to be the sole survivors of an ancient group of reptiles called anapsids, which lacked temporal openings in their skulls. However, molecular studies have consistently placed turtles within the diapsid reptiles, more closely related to archosaurs, crocodiles and birds, than to lepidosaurs, lizards and snakes. This placement of turtles within diapsids has significant implications for our understanding of reptile evolution. It suggests that the anapsid-like skull of turtles is a secondary adaptation rather than a primitive feature, and that the ancestor of turtles likely had temporal openings that were later closed during evolution. The fossil record has played a crucial role in refining our understanding of turtle phylogeny. The discovery of transitional forms like Odontocheles and Papacheles has helped to bridge the gap between early reptiles and crown group turtles, providing valuable information about the sequence of character acquisitions in early turtle evolution. Fossil discoveries have also revealed extinct lineages of turtles that don't fit neatly into the Cryptodira-Ploridira division. For example, the Mealoniidae, an extinct family of horned turtles from Australia and South America, have been difficult to place phylogenetically. Some analyses suggest they may be stem turtles, branching off before the Cryptodira-Ploridira split, while others place them within Cryptodira. The integration of fossil and molecular data has allowed researchers to estimate the timing of major events in turtle evolution. Current estimates suggest that the turtle lineage diverged from other reptiles around 260 million years ago, during the Permian period. The split between Cryptodira and Ploridira is thought to have occurred around 200 million years ago, in the late Triassic or early Jurassic. These dating estimates have important implications for biogeography, helping to explain the current distribution of turtle diversity around the world. For example, the early divergence of Ploridira and Cryptodira may explain why side-necked turtles are found primarily in the southern hemisphere, while hidden-necked turtles have a more global distribution. Recent phylogenetic studies have also shed light on the evolution of key turtle traits. By mapping characteristics onto phylogenetic trees, researchers can infer when and in which lineages specific adaptations evolved. This approach has been used to study the evolution of shell morphology, habitat preferences, and even physiological traits like temperature-dependent sex determination. The field of turtle phylogenetics continues to evolve rapidly, with new data and analytical methods constantly refining our understanding of turtle relationships. Future research is likely to focus on integrating multiple lines of evidence, morphological, molecular, and developmental, to create a more comprehensive picture of turtle evolution. As our understanding of turtle phylogeny improves, it not only enhances our knowledge of this fascinating group but also contributes to broader questions in evolutionary biology. The study of turtle relationships provides insights into the processes of adaptation, the impact of major environmental changes on evolution, and the complex interplay between genetics and morphology in shaping the diversity of life on Earth. 6. Convergent evolution in turtle-like forms. The evolution of the turtle body plan, with its distinctive shell and unique anatomical features, is a remarkable example of evolutionary innovation. However, what's equally fascinating is that similar body plans have evolved independently in other groups of animals, a phenomenon known as convergent evolution. Recent research has shed light on several instances of convergent evolution that have produced turtle-like forms in various lineages, providing valuable insights into evolutionary processes and the constraints and opportunities that shape animal body plans. One of the most striking examples of convergent evolution with turtles comes from an extinct group of marine reptiles called placodonts. These animals lived during the Triassic period, around the same time that the first turtles were evolving. Some placodonts, particularly the later forms, developed body armor that was remarkably similar to that of turtles. They had broad, flattened ribs that formed a carapace-like structure, and some even had a plastron-like ventral armor. The similarity between placodonts and turtles is so striking that early fossils of placodonts were initially mistaken for early turtles. However, closer examination revealed fundamental differences in the way their armor was constructed. While turtle shells are formed by the fusion of ribs and vertebrae with dermal bone, placodont armor was made up of bony plates that developed in the skin, more similar to the armor of crocodiles or armadillos. This convergence between placodonts and turtles is particularly interesting because it occurred in a marine environment, suggesting that similar selective pressures in aquatic habitats can lead to the evolution of turtle-like body plans. The protective value of a shell-like structure in an aquatic environment, where predators can attack from any direction, may have driven this convergent evolution. Another fascinating example of convergence comes from a group of Triassic reptiles called the Saurosphergidae. These animals, like placodonts, evolved broad ribs that formed a carapace-like structure. However, the Saurosphergidae took this convergence a step further. Not only did they have a turtle-like carapace, but they also had broadened gastralia, rib-like bones in the belly region, that formed a structure similar to the plastron of turtles. The similarity between Saurosphergids and early turtles is so striking that some researchers have suggested they might be closely related. However, detailed anatomical and phylogenetic studies have shown that saurosphergids evolved their turtle-like features independently. This example of convergence is particularly intriguing because it shows how even complex structures like the turtle's double shell system can evolve multiple times under similar selective pressures. Moving to more recent times, we find examples of convergent evolution with turtles among mammals. The glyptodonts, extinct relatives of armadillos that lived in the Americas until about 11,000 years ago, evolved a body plan that was remarkably turtle-like. These massive mammals, some of which grew to the size of a small car, had a domed carapace made up of hundreds of bony plates. Like turtles, their shoulder blades were located inside their rib cage, a highly unusual feature for a mammal. The convergence between glyptodonts and turtles is particularly interesting because it occurred in a completely different vertebrate lineage. This suggests that the turtle body plan is a highly effective solution to certain environmental challenges, one that evolution has discovered multiple times in different groups. Even among living animals, we can find examples of convergence with certain aspects of turtle morphology. For instance, the mata turtle of South America has evolved a flattened head and neck that allows it to blend in with the leaf litter on the bottom of streams and rivers. This adaptation for camouflage and ambush predation is remarkably similar to that seen in some species of freshwater stingrays, despite the vast evolutionary distance between turtles and rays. The study of these convergent forms provides valuable insights into evolutionary processes. It demonstrates that similar environmental pressures can lead to the evolution of similar traits, even in distantly related groups. This supports the idea that there are optimal solutions to certain evolutionary challenges, and that natural selection will often arrive at these solutions independently in different lineages. Furthermore, the study of convergent evolution can help us understand the constraints on evolutionary change. By comparing the similarities and differences between convergent forms, we can gain insights into which features are necessary for a particular way of life, and which are more flexible. The existence of these convergent forms also highlights the incredible plasticity of life. It shows that even highly derived body plans, like that of turtles, can evolve multiple times given the right selective pressures. This underscores the power of natural selection to shape organisms to fit their environments, even when it requires dramatic changes to body structure. As we continue to study these examples of convergent evolution, we gain a deeper appreciation for the complexities of evolutionary processes. Each instance of convergence tells a unique story of adaptation and provides a window into the interplay between environmental challenges and evolutionary responses. By studying these turtle-like forms across different animal groups and time periods, we enrich our understanding of evolution as a whole and the fascinating ways in which life adapts to the challenges of existence. 7. Evolution of turtle size and shape diversity. The remarkable diversity of turtle sizes and shapes we see today is the result of millions of years of evolution, adaptation, and specialization. From the tiny speckled padloper tortoise, which can fit in the palm of your hand, to the massive leatherback sea turtle that can weigh over a ton, turtles exhibit an extraordinary range of sizes. Similarly, their shapes vary from the high domed shells of some tortoises to the flat, pancake-like shells of soft-shelled turtles. Recent research has provided fascinating insights into how this diversity evolved and the factors that have influenced turtle size and shape over time. The evolution of turtle size has been a subject of particular interest to researchers. Early in their evolutionary history, turtles were generally small to medium-sized animals. However, over time, many lineages evolved towards larger sizes. This trend towards increased body size, often referred to as Cope's rule, is seen in many animal groups, but its applicability to turtles has been a subject of debate. Recent studies using fossil data and phylogenetic analysis have shown that the evolution of turtle size is more complex than a simple trend towards larger sizes. While some lineages did indeed evolve larger body sizes over time, others remained small or even decreased in size. This suggests that turtle size evolution has been influenced by a variety of factors, including habitat, diet, and evolutionary history. One fascinating example of size evolution in turtles is the case of island gigantism. On some islands, tortoises have evolved to be much larger than their mainland relatives. The most famous examples are the giant tortoises of the Galapagos Islands and the Aldabra Atoll, which can weigh up to 250 kilograms. This increase in size is thought to be an adaptation to island environments, where there are fewer predators and competitors, allowing the tortoises to grow larger over generations. Conversely, some turtle lineages have evolved towards smaller sizes. For example, the speckled padloper tortoise of South Africa, which reaches a maximum size of about 110 millimeters, is thought to have evolved its small size as an adaptation to living in arid environments where resources are scarce. The evolution of turtle shape diversity is equally fascinating. The basic turtle body plan, with its shell composed of a carapace and plastron, has remained remarkably consistent over more than 200 million years. However, within this basic plan, turtles have evolved an incredible diversity of shell shapes, each adapted to particular ways of life. One of the most striking examples of shape evolution in turtles is seen in the flatback sea turtle. Unlike other sea turtles which have a curved carapace, the flatback has an unusually flat shell. This shape is thought to be an adaptation to its habitat in shallow coastal waters, where a flatter shell may provide hydrodynamic advantages. Another interesting case of shape evolution is seen in the mata mata turtle of South America. This species has evolved an extremely flattened head and neck, along with a rough, bark-like texture to its shell. These adaptations allow it to blend in perfectly with leaf litter on the bottom of streams and rivers, where it lies in wait for prey. The evolution of shell shape in turtles has been influenced by a variety of factors. In terrestrial species, high domed shells often provide better protection against predators, as they are harder to crush. In aquatic species, flatter, more streamlined shells reduce drag while swimming. Some turtles, like box turtles, have evolved hinged shells that can close tightly, providing excellent protection against predators. Recent research has also revealed interesting patterns in the evolution of turtle shell shapes. For example, studies have shown that the shells of aquatic turtles tend to become more streamlined over evolutionary time, likely as an adaptation to improve swimming efficiency. Conversely, terrestrial turtles often evolve more domed shells over time, possibly for improved protection or to increase internal volume for storing water and food. The diversity of turtle sizes and shapes is not just a product of adaptation to different environments, but also reflects the long and complex evolutionary history of the group. Different turtle lineages have taken different evolutionary paths, resulting in the wide variety of forms we see today. For instance, the soft-shelled turtles have evolved a unique shell structure where the bony elements are greatly reduced, resulting in a leathery, pancake-like shell that is highly streamlined for swimming. Interestingly, the evolution of size and shape in turtles has not always been a gradual process. Studies of the fossil record have revealed instances of rapid evolutionary change, particularly in response to major environmental shifts. For example, the evolution of giant tortoises on islands appears to have occurred relatively quickly in geological terms, demonstrating the potential for rapid size evolution under certain conditions. The study of size and shape evolution in turtles has implications beyond just understanding turtle biology. It provides insights into broader evolutionary processes, such as how body plans can be modified over time, the interplay between form and function in evolution, and how animals adapt to different environmental niches. Furthermore, understanding the evolution of turtle size and shape diversity is crucial for conservation efforts. As we face global challenges like climate change and habitat destruction, knowing how different turtle species have adapted to various environments in the past can help us predict and mitigate the impacts of environmental changes on turtle populations in the future. As research in this field continues, we are likely to uncover even more fascinating details about the evolution of turtle size and shape diversity. Each new discovery adds to our understanding of these remarkable creatures and enriches our appreciation for the incredible diversity of life on Earth. 8. Turtle evolution in response to environmental changes. The evolutionary history of turtles spans more than 220 million years, during which time these remarkable reptiles have witnessed and adapted to numerous environmental changes. From the breakup of continents to dramatic climate shifts, turtles have repeatedly demonstrated their resilience and adaptability. Recent research has provided fascinating insights into how turtles have evolved in response to these environmental changes, offering valuable lessons about the adaptability of life and the long-term impacts of environmental shifts. One of the most significant environmental changes that turtles have faced over their evolutionary history is the breakup of the supercontinent Pangaea. This process, which began about 175 million years ago, had profound effects on the distribution and evolution of turtles. As continents drifted apart, turtle populations became isolated, leading to the evolution of distinct lineages in different parts of the world. For example, the split between Pleurodira, side-necked turtles, and Cryptodira, hidden-necked turtles, is thought to have been influenced by the separation of Gondwana, the southern supercontinent, from Laurasia, the northern supercontinent. This continental drift led to the predominantly southern distribution of Pleurodira and the more global distribution of Cryptodira that we see today. Climate change has been another major driver of turtle evolution. The fossil record shows that turtles have survived through periods of extreme global warming, such as the Paleocene-Eocene thermal maximum about 56 million years ago, as well as through ice ages. These climate fluctuations have had significant impacts on turtle distributions, body sizes, and adaptations. During warmer periods, many turtle species expanded their ranges towards the poles. For instance, fossils of tropical turtle species have been found in what are now temperate regions, indicating that these areas had much warmer climates in the past. Conversely, during colder periods, many species retreated towards the equator or became extinct in higher latitudes. Climate changes have also influenced turtle body sizes. Some researchers have observed a correlation between global temperatures and turtle body sizes in the fossil record, with many species becoming larger during warmer periods. This trend, known as Bergmann's rule in reverse, is thought to be related to the higher metabolic rates and increased food availability in warmer climates. The evolution of sea turtles provides a particularly interesting case study in adaptation to environmental change. The ancestors of modern sea turtles are believed to have been freshwater turtles that gradually adapted to more saline environments. This transition, which occurred during the Cretaceous period, involved not only morphological changes, such as the evolution of flipper-like limbs, but also physiological adaptations to deal with the challenges of marine life, such as the ability to excrete excess salt. The evolution of temperature-dependent sex determination, TSD, in many turtle species is another fascinating example of adaptation to environmental conditions. In species with TSD, the temperature of the nest during a critical period of embryonic development determines the sex of the offspring. This system is thought to have evolved as a way to optimize sex ratios in variable environments. However, it also makes turtles particularly vulnerable to rapid climate change, as rising temperatures could skew sex ratios and potentially lead to population declines. Turtles have also evolved various strategies to cope with changes in water availability. Some terrestrial species, like the desert tortoise, have developed the ability to store water in their bladders and reabsorb it when needed, allowing them to survive in arid environments. Conversely, some aquatic turtles have evolved the ability to absorb oxygen through their skin or the lining of their mouths, allowing them to remain underwater for extended periods in oxygen-poor environments. The impact of mass extinction events on turtle evolution is another area of active research. Turtles have survived several major extinction events, including the end Cretaceous extinction that wiped out the non-avian dinosaurs. While many turtle lineages did go extinct during these events, the group as a whole demonstrated remarkable resilience. Some researchers suggest that the turtle's shell and their ability to enter a state of dormancy during adverse conditions may have contributed to their survival through these catastrophic events. In more recent times, human-induced environmental changes have become a significant factor in turtle evolution. Habitat destruction, pollution, and climate change are putting many turtle species under pressure. While turtles have demonstrated remarkable adaptability over their long evolutionary history, the rapid pace of current environmental changes poses unprecedented challenges. Some turtle species are showing signs of rapid adaptation to human-altered environments. For example, some freshwater turtles have been observed nesting on artificial beaches or in agricultural fields when their natural nesting sites are destroyed. However, it's unclear whether most species will be able to adapt quickly enough to keep pace with current rates of environmental change. The study of how turtles have evolved in response to past environmental changes provides valuable insights for conservation efforts. By understanding how different turtle species adapted to past climate shifts, we can better predict how they might respond to current and future changes. This knowledge can inform conservation strategies, helping to identify which species might be most vulnerable and what measures might be most effective in protecting them. Furthermore, the long evolutionary history of turtles serves as a reminder of the resilience of life on Earth. Turtles have survived dramatic environmental changes over hundreds of millions of years, continuously adapting and evolving. While the current rate of environmental change poses significant challenges, the evolutionary history of turtles gives us hope that, given the chance, these remarkable creatures can continue to adapt and thrive. As we continue to study turtle evolution in response to environmental changes, we gain not only a deeper understanding of these fascinating animals but also broader insights into the processes of adaptation and the impacts of environmental change on life on Earth. Each discovery in this field enriches our appreciation for the complexity and resilience of life and underscores the importance of protecting the diverse ecosystems that support it. 9. Molecular and developmental insights into turtle evolution. The field of turtle evolution has been revolutionized in recent years by advances in molecular biology and developmental genetics. These new approaches have provided fresh insights into the genetic basis of turtle traits, the developmental processes that shape the unique turtle body plan, and the evolutionary relationships between turtles and other reptiles. By combining these molecular and developmental perspectives with traditional paleontological and morphological studies, researchers are gaining a more comprehensive understanding of turtle evolution. One of the most significant contributions of molecular biology to the study of turtle evolution has been in clarifying the phylogenetic position of turtles within the reptile tree of life. For many years, the evolutionary relationships of turtles were hotly debated, with some researchers suggesting they were the sole survivors of an ancient group of reptiles called anapsids, while others argued they were more closely related to diapsid reptiles like lizards and crocodiles. Molecular phylogenetic studies, which compare DNA sequences across different species, have consistently placed turtles within the diapsid reptiles, most likely as a sister group to archosaurs, the group that includes crocodiles and birds. This finding has significant implications for our understanding of reptile evolution, suggesting that the anapsid-like skull of turtles is a secondary adaptation rather than a primitive feature. These molecular studies have also helped to resolve relationships within turtles, confirming the basic split between Ploridira and Cryptodira and clarifying relationships between various turtle families. They've even provided estimates for the timing of major evolutionary events in turtle history, such as the divergence of different lineages. Beyond phylogenetics, molecular studies have provided insights into the genetic basis of key turtle traits. For example, researchers have identified several genes involved in the formation of the turtle shell. One key player is the WNT signaling pathway, which is crucial for the development of the carapace. Genes in this pathway are expressed in a unique pattern in turtle embryos, helping to direct the growth of the ribs and their fusion with the dermis to form the shell. Another important gene in turtle shell development is gremlin, which is involved in limb development in other vertebrates. In turtles, gremlin is expressed in a novel pattern that correlates with the development of the carapacial ridge, a key structure in shell formation. This finding suggests that the evolution of the turtle shell involved the co-option of existing developmental pathways for new functions. Developmental studies have also shed light on how turtles evolved their unique body plan, with the shoulder blades positioned inside the rib cage. This unusual arrangement arises during embryonic development through a novel pattern of cell movements and gene expression. Understanding these developmental processes helps explain how such a dramatic anatomical change could have evolved. The evolution of temperature-dependent sex determination, TSD, in many turtle species has also been a focus of molecular and developmental research. Studies have identified key genes involved in this process, such as CIRBP and TRPV4, which are sensitive to temperature and can influence sex determination. Understanding the molecular basis of TSD is crucial for predicting how turtles might respond to climate change, which could affect nest temperatures and thus population sex ratios. Comparative genomics, which involves sequencing and analyzing entire genomes of different species, is providing new insights into turtle evolution. For example, Analysis of turtle genomes has revealed that turtles have a particularly slow rate of molecular evolution compared to other vertebrates. This slow evolutionary rate might be related to their long lifespans and low metabolic rates. Genome studies have also identified several genes unique to turtles, as well as changes in gene regulation that may be responsible for some of their distinctive features. For instance, researchers have found changes in genes related to bone formation that may have been important in the evolution of the turtle shell. Epigenetic studies, which look at how environmental factors can influence gene expression, are providing insights into how turtles adapt to different environments. For example, studies have shown that the temperature during embryonic development can affect the expression of genes involved in metabolism and immune function, potentially influencing the fitness of turtles in different climates. The field of evolutionary developmental biology, or EvoDevo, has been particularly fruitful in understanding turtle evolution. By comparing the embryonic development of turtles with that of other reptiles, researchers can identify the developmental changes that led to the unique turtle body plan. For instance, studies have shown that the turtle shell develops from a novel embryonic structure called the carapacial ridge, which is not present in other reptiles. These molecular and developmental approaches are also shedding light on the evolution of other turtle traits, such as their longevity. Turtles are known for their long lifespans, and researchers have identified several genes that may contribute to this trait, including genes involved in DNA repair and cellular stress responses. As technology continues to advance, new molecular techniques are being applied to the study of turtle evolution. For example, ancient DNA techniques are allowing researchers to extract genetic material from fossil turtles, providing direct evidence of genetic changes over time. Meanwhile, CRISPR gene editing technology is opening up new possibilities for experimentally testing hypotheses about gene function in turtle development. The integration of molecular and developmental approaches with traditional paleontological and morphological studies is providing a more comprehensive picture of turtle evolution than ever before. This multidisciplinary approach is not only enhancing our understanding of how these fascinating creatures evolved but is also contributing to broader questions in evolutionary biology, such as how novel body plans arise and how organisms adapt to changing environments. As we continue to unravel the molecular and developmental basis of turtle evolution, we gain a deeper appreciation for the complexity of evolutionary processes and the remarkable adaptability of life. Each new discovery in this field brings us closer to understanding the full story of turtle evolution, from the genes that shape their unique features to the developmental processes that build their distinctive body plan. Key takeaways and broader implications. As we've journeyed through the fascinating world of turtle evolution, we've uncovered a wealth of insights that not only deepen our understanding of these remarkable creatures but also shed light on broader evolutionary processes. Let's take a moment to synthesize the key points we've explored and consider their wider implications. First and foremost, the study of turtle evolution provides a powerful example of how major anatomical innovations can arise through a series of incremental changes over millions of years. The turtle shell, one of the most distinctive features in the animal kingdom, didn't appear overnight. Instead, it evolved gradually, beginning with the broadening of ribs in early ancestors and culminating in the fully formed carapace and plastron we see today. This underscores the power of natural selection to shape complex structures over time, a principle that applies across the tree of life. The convergent evolution of turtle-like forms in other lineages, such as placodonts and glyptodonts, highlights the fact that similar environmental pressures can lead to similar adaptations, even in distantly related groups. This principle of convergent evolution is a fundamental concept in evolutionary biology, demonstrating that there are often optimal solutions to particular ecological challenges. The diversity of sizes and shapes we see in modern turtles, from tiny terrestrial species to massive sea turtles, illustrates the adaptability of the basic turtle body plan. This diversity is a testament to the power of evolution to modify existing structures to suit a wide range of ecological niches. It also serves as a reminder that evolution doesn't always proceed in a straightforward direction towards larger size or greater complexity, but rather optimizes organisms for their specific environments. The ability of turtles to adapt to major environmental changes throughout their long evolutionary history, from the breakup of continents to dramatic climate shifts, demonstrates the resilience of life. This resilience offers a glimmer of hope as we face current environmental challenges, suggesting that given enough time and opportunity, species can adapt to new conditions. However, it also underscores the importance of conservation efforts to ensure that species have the time and space they need to adapt to rapidly changing environments. The insights gained from molecular and developmental studies of turtles have broader implications for our understanding of evolutionary processes. The co-option of existing developmental pathways for new functions, as seen in the formation of the turtle shell, is a common theme in evolution. This highlights the often opportunistic nature of evolutionary change, where existing structures and processes are repurposed rather than invented from scratch. Moreover, the study of turtle evolution contributes to our understanding of major evolutionary transitions. The move from land to sea in the evolution of sea turtles, for instance, provides insights into how organisms can adapt to radically different environments over time. This has implications for our understanding of other major transitions in the history of life, such as the evolution of flight or the transition of mammals back to aquatic life. The longevity of turtles and their ability to survive multiple mass extinction events offer intriguing avenues for research into aging and resilience. Understanding the genetic and physiological mechanisms that contribute to turtle longevity could have implications for human health and aging research. From a conservation perspective, the study of turtle evolution provides crucial insights. Understanding how different turtle species have adapted to past environmental changes can help us predict and mitigate the impacts of current climate change and habitat destruction on turtle populations. This knowledge is essential for developing effective conservation strategies to protect these ancient and remarkable creatures. Finally, the ongoing debates and discoveries in the field of turtle evolution remind us that science is a dynamic, ever-evolving process. New fossil discoveries, advanced analytical techniques, and innovative research approaches continue to refine and sometimes revolutionize our understanding. This serves as an inspiring reminder of the excitement of scientific discovery and the importance of remaining open to new evidence and ideas. As we look to the future, the study of turtle evolution will undoubtedly continue to yield fascinating insights. Each new discovery not only enhances our appreciation for these remarkable animals but also contributes to our broader understanding of evolutionary processes, the history of life on Earth, and the intricate relationships between organisms and their environments. In conclusion, the evolutionary journey of turtles, spanning over 220 million years, is a testament to the power of adaptation, the resilience of life, and the wonders of evolutionary processes. As we face global challenges like climate change and biodiversity loss, the lessons learned from turtle evolution can inform our approaches to conservation and environmental stewardship. By understanding and appreciating the remarkable evolutionary history of turtles, we gain not only scientific knowledge but also a deeper connection to the natural world and the countless species with which we share our planet. Thank you for joining us on this exploration of turtle evolution. We hope this journey has sparked your curiosity and deepened your appreciation for these fascinating creatures. If you've enjoyed this video, please consider subscribing to our channel for more in-depth explorations of evolutionary biology and natural history. Don't forget to like this video and share your thoughts and questions in the comments below. Your engagement helps us create content that resonates with our audience and furthers scientific understanding. Until next time, keep exploring, keep questioning, and never stop learning about the wonders of the natural world.